Hi everyone, welcome all to the SciChat episode 4. That is, we'll be discussing on antibiotic resistance. We'll be talking about evolutionary biology, perspective in bacteria, and how the bacterium develops antibiotic resistance, and as the title says, penicillin to pandemic. So there have been many pandemic, bacterial pandemic, even in India, abroad, and in the world, like cholera, tuberculosis, and many more uh, in past history. So I think it's a major thing and a global issue now that we need to really talk upon. And for that, I would say we have a evolutionary biologist who is currently working in the field of antibiotic resistance. Not currently, I would rather say working since a long, at least I would say 5 to 6 years now. No,
4 years. Yeah, 5 years
4 years.
would be too
Okay,
much. Yeah.
4 years, at least 4 to 5 years
Yeah.
of antibiotic resistance. And now Mr. Surajit, he is doing his PhD at University of Kiel, and he's also affiliated to Max Planck Institute of Evolutionary Biology, which is located at Kiel. So we have a pleasure of listening to him today, and also you all joining today. So I would like to start this session, and it's a Sunday morning, and thanks everyone for joining, right? And especially the people who are in Europe right now. It's morning 8 a.m. for
Exactly.
Surajit and me, and it's quite cold. It's like I
Exactly.
think
Yeah.
it's 10 degrees right now.
Yeah. It's been
So
freezing cold here
so
for
yeah,
last
yeah,
couple of
yeah,
weeks.
yeah,
Yeah.
yeah, yeah. So yeah, so thanks for joining everyone. So let's quickly start upon. I'll ask you a very simple question to everyone, and I hope we keep this iChat interactive. And I see many familiar faces today. Many.
Yeah. Me
I
too.
would say
Me too.
I
Definitely.
could see
Yeah.
many familiar faces today, which I have maybe seen them earlier, sometime in SciChat, 2, 3 years back, maybe some now as well. So it would be really good if you guys can also turn on your cameras so that uh, we can interact personally. So those who wants to turn on your cameras and uh, video, audio, you are all free to do it, because rather to be a two-way conversation, it would be really good if we have like many people joining, right? That's the goal of the side chat. So yeah, okay. So I would like to start this session with a very small question. So consider that you have a flu, okay? Irrespective that you don't know now, you have not been diagnosed that you have a flu. What is the cause of that? Which antibiotic would you prefer to take? Maybe you can write it in chat box. Which antibiotic do you take usually? I think I'll give 1 minute and drop down in chat box quickly, or maybe you can speak up like citizen, PCM, or what. No, citizen and both, they both are not antibiotic. Yeah. But which antibiotic do you take? Amoxicillin.
Yeah. Amoxicillin, tetracycline,
Yeah.
any beta lactam, if you think so.
Let people say, I would
Okay.
say.
So I got a response
So
from
please write
Gmail.
down in the chat box
It's
that which
ciprox.
antibiotic do you take
Ciprofloxacin.
if
Okay.
you have a flu.
So he's telling like for a common cold, he might go for ciprofloxacin. I can see
Uh-huh.
some
Uh-huh. Yeah, yeah,
hand.
yeah. Yeah, go ahead, go ahead. Yeah, I think you don't need to raise your hand in this
Yeah,
meeting. You
please
can just
just pick
speak up.
up,
That's
please.
fine. Yeah.
Yeah.
I don't know who was
May I know
that.
the reason why did you say ciprofloxacin? Sorry, if you're not really comfortable, or if you're just guessing, or if
Yeah,
it's just a blind
like
guess, then also it's fine.
yeah.
Okay.
Yeah. So you can speak up. Okay.
Mm-hmm.
Okay. So Surajit,
Citrin,
citizen
citrin
is not
is not an antibiotic.
antibiotic,
It's
but
a
yeah,
antihistamine tablets, which actually,
antihistamine.
yeah,
Yeah,
which blocks the hypersensitive
correct.
reactions. Yeah. So now, in common cold, you should not take any antibiotics because it's caused by virus, and the virus has actually, if you take antibiotic, that particularly kills bacteria. So that is the misconception that
Yeah,
we have been hovering around for almost like at least 20 to 30 years. Like, we definitely do not know the reason why do we take antibiotics, and sometimes it also works like a placebo effect, because
yeah,
a patient just feels like, okay, if I take antibiotics, it might be all good. So exactly, Somalia. So common cold is caused by rhinovirus, and the rhinovirus is a virus, and we know antibiotic has a selective specificity for bacterial cells because it has been designed in such a way so that it could actually kill bacteria. So if you administering something which is not really meant for that particular pathogen, it's of no use, right? So why do you take
yeah,
that? So these are the
yeah.
misconceptions. So
Yeah. So I start with this question because most of them take antibiotic like eating like badam or kaju. Few of them
yeah,
really do.
exactly.
Kuch hua, you just take antibiotic. But this is not really what it is. And antibiotic are taken and at some severe bacterial infections that
Hmm.
to if you diagnose them correctly and you the doctor prescribes a correct antibiotic. So there were so many antibiotic being taken in COVID times, and I was wondering why. In fact, antibiotic will lower down your immunity somehow,
Hmm.
and antibiotic do does because it disturbs the gut microbiome. Like,
Hmm.
I don't know, have you guys noticed it or not? At least I have noticed it with me. But Surajit, maybe you can correct me.
Hmm.
Uh, when you take a high dose of antibiotic,
Hmm.
even your gut microbiome washes away, and then
Definitely.
you have a diarrhea effect on later day when you have a heavy dose of antibiotic.
Definitely, Dharia.
I'm
It
yeah,
does really produce some effect on your gut microbiome,
yeah,
because if you are giving a selection pressure, like as an evolutionary microbiologist, I love to think in this direction. If you are
yeah.
giving an antibiotic, that means you are imposing a selection pressure. And if you are
Hmm.
giving a selection pressure to a community, of course, you are actually selecting from for some resistant variants. So as a result, the normal microbiota are getting resistant toward antibiotics. So knowingly or
Yeah,
unknowingly, you are actually making yourself resistant to antibiotics
correct.
because of taking them. Now, what's really unfortunate these days, like the mothers, like those, like the pregnant lady, and also, for example, those are really treated with antibiotics in the very first phase of the pregnancy. We also often see that there is a chance, like the microbiota that has been got dysbiotic from the mother, and it's been actually transferred to a child. So it's not, you are not only damaging yourself, on the other hand, this has also been channelized to your child, and it goes around. And as you know, this is an evolutionary arm race where host evolve, parasites evolve. So it actually, you are making them more resistant far away. So it's like, it definitely, it creates a lot of
I
effects
see.
on your gut microbiome. Yep.
Yeah, definitely. Okay, so Surajit, now let's start this event. You shared a lot of things in the start itself, and now I guess people are motivated enough. So, Surajit, maybe you can quickly introduce yourself and give a short glimpse that what was your research journey,
Hmm.
and how do you reach here wherever you are right now,
Hmm.
and yeah, and why specifically you choose AMR
Hmm.
as your research topic, and what excites you about AMR?
Okay, thanks, Dharia, for conducting this session. So first, I would like to take this opportunity to welcome you all for joining this Sunday morning, because we know this weekend has always been a bit where people should take rest, not really rather discussing about science. It's not only it's not a good practice enough. So, but you really, all of you are really motivated enough, I would say, to help us joining this event, which definitely makes this even more successful. So, thanks for joining again. And the second thing, I could see a lot of familiar names. So thanks to for all these platforms like Zoom and Google Meet, where you could actually get a chance to meet people virtually if you are not really present in person. So my name is Surajit Pal. I completed my bachelor's in microbiology back in 2018. Then I moved to Pondicherry University to pursue my master's in microbiology. I also had a privilege to do a diploma in biotech. That was a kind of an add-on course which I attended, and there I also made some good friends like Ankit. He is also here with us today. So like, and then I actually, it was like very unfortunate for all of us because like COVID pandemic 2020. So we definitely didn't have any clue what to do next. So I naively chosen for some of the internship, and those were online. And that time it was, if I remember correctly, the all the internship was offered by NEIST. That's a SRTP program. I think most of you have already done it during that time. So it was like a summer research training program organized by some institute in Jorhat, Assam. So then I worked with Bhavna Muralidharan at InSTEM as a part of it. And that really motivated me to work in the domain of gut microbiome dysbiosis and its correlation to brain cognitive functions. Uh, it was really then I tried to focus on my particular area of interest, but I definitely didn't have any clue about my own interest because it sometimes it takes a while to think of, like which actually excites you most. So I worked as a junior research fellow in Central University of Kerala, but it didn't really turn out to be a good one, of course. Then I just left my job, a lot of things behind undone. I'm not really going to explain why did I leave my job because it's not a good platform to discuss all the scientific controversies, and I will just keep it very simple as it is. So what I did, and that really motivated me to go further, was I worked with a fantastic professor, I would say, and he was like a guru to me. His name is Dr. Indran Chamato. He is based at West Bengal University of Animal and Fishery Sciences. It's sponsored by Indian Council of Agricultural Research. So where he actually told me directly, Shurajit, I may not really offer you any fellowship, even you clear gate and all. It's totally fine, but I can't really offer you any fellowship. But what I can offer you some publication. If you work on this bioinformatics approach, I can guarantee that I will give you some publication. And for me, on particularly that time, it was like, wow, I can't really expect more. I don't need any money because I'm staying in Calcutta and I can definitely go to your institute just to work on this particular topic. And I know about my bioinformatics stuff and I can just do it remotely. I don't have to travel every day. Then I joined ICR Institute in Calcutta and I started applying abroad. Of course, it's like Dharian knows and Anupam Da also knows it's not really so easy to get a position in abroad. You get at least 20 to 30 rejections day one after another, and it really feels like sucks. Like, oh man, where I am? Do I really belong here? Then I got an opportunity in Taiwan, but my family were not really so happy. I'm not really creating any bias here, like Taiwan is not a good country because Europe is rather more big country or something. But no, but I was not really so happy because I don't want to shift my field to agriculture because I was a microbiologist by my training. So I just wanted to do something with bacteria, but it was not really that has to be antibiotic resistance. So I just gave it a try to this professor, Henry Schulenberg, where I'm working now, my PI. I thought it's never going to be happen because it's like a Max Planck Institute, and we know like the Max Planck Institute comes under the top research clusters in the world. But I thought, let us just give it a try. Then I got shortlisted, and then things really worked out in a proper way, and I'm here today. So yeah,
That's really great. Yeah, Surajit, I would say
thanks.
it is a very fantastic journey that you have been through a lot of research experiences
Hmm.
before you reach here. And I would say it is much more important, to gain research experience, because
Definitely.
right now, when I go to my lab, either there is someone who is working on their own pace, or maybe independently. So everyone have to work independently abroad.
Definitely.
So you cannot expect help from even your colleague or your PhD lab mate who is doing a PhD
I
with
totally
you.
agree. Yeah. Hmm.
So it's very important that you have a relevant research experience. That's what I always tell to everyone, that if you're planning to move abroad,
Hmm.
make sure you have a relevant research experience, that if you are given something,
Maybe I'd like to
you
just
can
add
do
a
it for
single,
yourself.
very short point. If I don't know my audience, to be honest, but if you are a student and if you are doing research and your dream is to pursue your PhD in abroad, try to publish an article, not a
Yeah,
review or chapter. That may not
yeah,
really help you to get a position
right.
abroad because no one really cares if you publish a chapter or review.
Yeah, yeah,
Even we have a problem here. The moment I told my boss, Henrich, is that I have a chapter, he was so surprised to know how can you write a chapter because you are not an expert. And that
yeah,
is the dogma we are actually facing these days. Like
yeah,
we are writing a chapter, but we are not expert in that. And if you do
exactly.
that unknowingly, please stop it because it will never give you any opportunity to
Yeah,
work in abroad. Instead of that, you work on your bioinformatics, you do something in statistics, you publish a paper. So an article can give you a position in abroad for sure, but a chapter review may not really complement. It might
yeah,
help you to some extent. Nothing is better than anything. So it's like, but it's of no use again. So yeah.
yeah. To be honest, like when I applied for my PhD here abroad, last year in 2022, I think we were on the same
Hmm.
line
Yep.
track, Surajit.
Exactly.
When you got a position, even I got the
Hmm.
position, and we shifted here together, maybe a difference of
Definitely.
one
Yep.
month. I came
Yeah.
here
Yeah. Yeah.
in
I
August
think
end, and you came in the September end.
I
So
joined the fall semester. Yeah, I joined fall
yeah, so
semester.
even I joined the fall semester. It was maybe
Mhm.
only 15-20 days that
Yeah.
and we were discussing about the visa issues as well, I
Yes, yes,
remember.
yes, exactly. I still remember that.
Okay,
Yeah.
okay, okay. So now let's come back to the topic. Lot of
Sure.
PhD talk. So Surajit,
Yeah.
I would now like to ask you few questions which are very much relevant to AMR and audience that joined today for this discussion, especially. So, being a researcher,
Mm-hmm.
what do you feel that what are the research gaps that are prevailing in the AMR region right now, and how do you identify your domain? I would say, especially in the field of antibiotic resistance, and talking about in the evolutionary perspective of bacteria. Uh, what are your what research gaps did you find out during
That's a
your last 5 years?
that's a very clever question there. Yeah, but basically in India, if you could see, or also in abroad, people are working a lot with AMR. But I think the major part which is missing from their research is the evolution. Now, evolution is something is the key of antibiotic crisis because bacteria having a very short generation time, it evolves way more faster than any other organism you could actually think of. And it is still there from the very beginning when it all started. Like the life started in Earth, and the bacteria were still maintaining like a single cell from that to now today onwards. And it has no effect, like it has no changes. So why are they still going to pursue in this direction? So that make them evolve, so they could actually evolve way more faster than any other organism. And if we are not really considering those evolutionary dynamics, that is missing. It's really unfortunate to see these days. A recent publication came in 2016. It's an article published by Lancet where we at the Schulenburg lab, we just did a very simple literature survey. What we did, sorry, it's like a keyword survey. We just opened the article, Control F, and we typed evolution. It was so unfortunate for us, not even a single word was mentioned there about evolution. Of course, there was some word, it was mentioned like development, but those developments are like developing new antibiotics. But you need to keep in mind, if you are also going to develop a new antibiotics, it's just a span of 1 to 2 days. Because I am really sure when I'm saying 1 to 2 days, because I can actually show you in my today's presentation, like bacteria are way more faster than any other organism, and it evolves so fast that actually in a clinical treatment, if you give an antibiotics, it will evolve and come out with a new resistant variant so fast. So I think this evolutionary dynamic needs to be taken into the account if we are, really thinking of something called a sustainable therapy. Because if you really want to make a treatment which has to be a sustainable for your patient, you need to really consider evolution. I can definitely show you how exactly are we working on it at the Max Planck Institute and also the university. So
Mhm.
yeah, maybe not now. Then I will just keep it secret
Yeah,
for the
yeah,
time being. Yeah.
definitely. Yeah, even I feel so because I am a not full time virologist. I am a
Hmm.
molecular biologist, but since I deal with virus, I understand the amount of mutation rate that even virus have, and also the more important thing, the mutation that we want to create through
Hmm.
CRISPR-Cas9 or Trilog system in the viral DNA or
Hmm.
RNA, depending upon whatever virus we would be working. So based upon that, we want some mutation and we get it
Exactly.
some else.
That is the mistake, and often you find, like, if, for
Yeah,
example, the most common problem, Harry, what is like mutation is something, and the point mutations, they are really nasty. So if you do an experiment, the next morning you are coming and you could just see those point mutation will heck you up a lot, and
yeah,
then you have to really rethink to
yeah,
what to design, because the CRISPR-Cas9 is definitely a fascinating system. You
yeah,
could definitely work on that, and you
yeah,
could design some innovative therapies. But
yeah,
nowadays, people have already reported
yeah,
even the CRISPR binding site is also being bacteria started modifying it, and
yeah,
they will do that. Everyone wants to live. Who
yeah.
wants to die in this universe? No one, right?
No one.
So they would try to develop a strategy that might combat them to it, like
Yeah,
fight against infections, and that's what
yeah.
they're doing. So yeah.
But to be honest, I am working on the cell death, so I want cell to die.
Oh yeah, of course.
but
Like
with a program, but
yeah,
in a
but
way that it
our ultimate,
programs its cell death, not a necroptotic cell death that usually bacteria and virus infection does when they are done with their work. But we are hoping to
but
make
those
some
are
specific
definitely some special cell are not they, right?
epithelial cells.
Yeah, exactly. So it's fine.
Epithelial cells, they don't die easily even
Hmm.
when the virus hijacks them.
Okay.
And immune cells are the most, they die like within 24 hours,
Mm-hmm.
that they are gone.
Great.
So yeah. So okay, so you talked about that evolution is missing in the antibiotic resistance right now, and that is a
Exactly.
major gap in the current antibiotic
Crisis.
resistance field. So
Mm-hmm.
so you are I would like to ask you one more question on this, that you told that bacteria just evolve so quickly in 1 or 2 days, and you
Mm-hmm.
also have some experimental proofs to it,
Mm-hmm.
right? So I would like to ask that you agreed on this beautiful title of today, that is penicillin to pandemic. So, what do you think as an evolutionary biologist? Because I have heard, I read some articles in Speaking Tree of Times of India and some other blog spots as well, where they have written that every 10 or 15 years, evolutionary per se, there is some pandemic
Mm-hmm.
that comes. So I don't know. There are many. Uh, there were many misconceptions. Talking evolutionary,
Mm-hmm.
that okay, this much amount of species would be first degraded, or 25% amount of species would be gone, and then when again it develops, and then there would be a pandemic again, and so that the
Mm-hmm.
evolutionary per se it goes on. So would you like to comment on this, that are we near to a second pandemic soon, or
Definitely, if we are not really careful enough by this time at least, because the most problem, in the hospital infections like the nosocomial pathogens, if you don't have any antibiotics to treat them, so you are running out of any options then, because your patients are sick, and unfortunately you have to give some medicine as a doctor. And if you are saying I don't have any medicine to give you, so then the patient will say, then should I die? Because it's like a kind of like, we are now like our discovery of this, like the pipeline of developing a new antibiotics has somehow now got stagnant. We are not really coming up with a new antibiotics. There is also a potential arguments that we could find out these days. If you are creating a larger molecule, for example, antimicrobial peptides, bacteria often face a lot of difficulties to digest them, and also it's really hard for them to develop resistance. The another approach, what I would love to think as an evolutionary microbiologist, is that you come up with a more synchronized protocol where you give antibiotics for a particular time, and then immediately you stop antibiotic and start strengthening up your inner gut microbiota. As a result, if you could just do these two things on a very complementary basis, I think that might help you to get yourself, like to get yourself improve in a more faster way than any other conventional method that we had been actually hovering about for almost last 20 to 30 years. It's like you have to complete your antibiotic dosage. I'm not saying, and I'm not really against it, like you don't have to complete your antibiotic dose. One should definitely consider that. But instead of that, in the Schulenberg lab, what we could also experimentally have already seen, if you are not really also completing your antibiotic dose and you're stopping your antibiotics, and at the same time, if you're just trying to, imposing some of the good microbiota, so the one that you, because antibiotics is like a very blind person, it doesn't know what's good and what's bad. It's killing both your beneficial microbiota, both the pathogenic one. So if the pathogenic one is are getting killed, that's good for us, then the infection load is getting down. But if the good microbiota, what actually helps us to, like synthesize some of the nutrients, some and also help us to digest vitamins and all other components which are really necessary for our cells and for our body for an overall nourishment. And if they are all getting died, it's a definitely problem. So now the pandemic, we just tell it on a very tricky way, because often the antibiotic resistant infections are life threatening. And if you have a life threatening infection, if you don't have a medicine, people will die. So it will be a pandemic. I
Does
think
it
I'm
doesn't
getting a
it
lot of,
sounds crazy, Surajit?
definitely it is. And it's also very
So
scary. I think,
so we have a question
yep,
from Kavil.
yep.
He asks that speaking of pandemics,
Hmm.
why does it seem that pandemic events occur in a cyclic manner? So
Yep.
there is some pattern that he has observed. Last major one was Spanish flu in 1918. Now Corona in 2019. Okay, I think Surajit is much more expert on this.
Honestly, that was a really fascinating to me even to see, because, but I still say I don't have a good explanation for it. But mm, I also wouldn't say it's a coincidence. But you see, any time for even for a virus to come up with a new mutation that has been actually going to be a more detrimental effect for a human cell, it needs a time. And also it has to jump from one host to another retrospectively. And then the moment it does it, it actually strengthens, for example, what we have seen from the history. Even these are actually very fascinating topic these days. We call it life history traits. So the life history trait, what it says, if a parasite, because this is ultimately host parasite interaction, you consider host human and the parasite is COVID-19. So the moment it actually came from a bat, like, to a human origin, it might have already surpassed by a lot of other hosts. And the moment it has been encountered to other hosts, it actually got some beneficiary mutants for them. It was beneficiary for them, not for us. And it was deleterious for us. So it actually takes that time. But I'm not really absolutely certain about it, like why there is 100 years of gap. It could sometimes more than that. Even if it's a bacterial pandemic, I can assure you it may not be 100 years of gap because they evolve so faster than any other virus as well. And the most intriguing part, virus needs a host system to survive. Bacteria does not, because it can survive independent, right? Because as you all might be knowing, virus is absolutely inert when it's out of your body. So in order to mutate itself, it needs to complete its life cycle in a host. But in bacteria, it does not. So it doesn't really
Yeah,
care. So
yeah,
so we
yeah.
have to,
I think we'll come on that,
sure,
Surajit.
sure,
We
sure.
will come on that with the relevant topic. So yeah, yeah, people, you guys
So,
can
but
just post
coming to
your
your answer,
questions.
Kabir, I'm not really sure why this 100 years of gaps were necessary in order to get the second pandemic. But
Hmm.
it takes a time to
Hmm.
develop some mutations for a
Hmm.
virus, which might
Hmm.
be detrimental for us.
Hmm.
So it definitely takes the time.
Hmm.
Yep,
Hmm. I see.
yep.
I see. Okay. So yeah, like now let's move ahead in this discussion, and like we are going into really interesting phase now.
Hmm.
Uh, so, Surajit, I would
Yep.
like to ask you that how bacteria develops, antibiotic resistance per se,
Hmm.
and can we do something, to stop this process somehow
Hmm.
in the bacteria first?
Okay.
And what are
Hmm.
your comments on this? And like, what would you like to tell first on the first half of the question, that is, what are the basic molecular mechanism? How does a bacteria develops antibiotic resistance?
I
Speaking
would, yeah.
molecularly. Yeah.
I would actually. There are a lot of papers available online. I know the people, those who have already joined, may definitely are from the background of antibiotic resistance. So it
Mm-hmm.
wouldn't be a good way to, if I just start saying, these are the methods, one, two, three,
Uh-huh.
four. It may
Uh-huh.
not be a good idea, but I would
Mm-hmm.
definitely try to point out a few of the fundamentals that we actually consider to be a key player in developing resistance. Is that the
Mm-hmm.
first one, bacteria, if it produces mutation to its target, those targets are really necessary for an antibiotics to go and bind. For example, you have an amino acid tRNA site, you go and you know that like the bacteria knows that antibiotic will come and bind here. So what if it just make the site a bit bulging in, and or else if it just modify that target site so that your antibiotics may not really allowed to sit there and it will get dislodged from that area. As a result, bacteria will achieve resistance. We could definitely see that for an antibiotics for like aminoglycosides, for example, per se. When it comes to some of the most prevalent resistant determinants, I would definitely say efflux pump. Efflux pump is a very simple pumping mechanism, what you could consider as like if you have some toxic substances inside your cell, you just kick it out. You just pump out it again, and then you just throw it outside. And it definitely does not really take much time to come up with an efflux pump machinery. Even every bacteria, whether it's a gram positive or gram negative, it has got an efflux pump. And there are tons of reviews and literature which definitely claims like the importance for this pump, not only for antibiotics, even sometimes it also helps them to carry out some of the other toxic substances that bacteria produces, right? So as a third method, what actually we are facing, like XDR, that is extensively drug resistant in MDR pathogen, because of we have certain enzyme that cleaves or that damage the antibiotic. As a result, your antibiotics may not really work because if you have an antibiotic called beta lactam, so this beta lactam antibiotic actually cleaves the beta lactamase linkage of particular, like the enzyme produced by beta lactamase actually cleave the beta lactam antibiotic. So if the beta lactamase ring is cut off, then of course your drug may not really work, right? So bacteria are producing those enzymes. We could see them a lot when it comes to ESBL, and there are also a lot of variants, and they are also evolving very faster than actually we could think of. Even nowadays, thanks to AlphaFold, because they are doing a groundbreaking discovery with predicting actual mutation and their effects on a protein level. Because it's like one of the most fascinating things in biology could be how a particular protein folds, because we know based on those folding patterns, the whole function will change. So the way how tactfully a particular protein is getting folded and how an artificial intelligence could be used as a tool to predict that, it itself is a very fascinating thing to observe. So but also that's not really an answer to the question that you have asked. And what was the second part of your question? I'm sorry, I'm sorry.
So yeah, you very well answered the first part of the
Mm-hmm,
question, Surajit. So what I understand and what I can summarize is that
mm-hmm,
there is an antibiotic. Okay, it's generally, I guess, it's a peptide or it's a protein. I'm not
mm-hmm.
sure. Or like, or what is the molecule of antibiotic first? Let's talk about this. What is an antibiotic?
Okay, okay.
Is it a
Yep.
protein molecule or is it a
Mhm,
glycoprotein molecule or like how would you call it as an antibiotic? Okay, it has a target site in the
mhm.
bacteria, maybe on the membrane so that it ruptures the membrane, some membrane protein, or maybe it targets some cytosolic
Mhm.
protein or cytosolic molecule. But what are antibiotics? Like, are they protein molecules or what?
I think it's a very fundamental question, and it's also necessary for us to understand. So, antibiotics are the substances that
Mm.
either kill or inhibit bacterial growth.
Okay.
So, as an evolutionary process, I would say bacteria, when it fights with each other, they produce antibiotic as a defense mechanism. So it is not something, recently coming up as a resistance problem, because
Hmm.
in past, if you could see, you have a soil. For example, I am giving you a beaker with 10 gram soils in it, and you would get a lot of bacteria in it. So on those bacteria, one bacteria will definitely try to kill others because of the space issues and also nutrient depletion. So they would try to fight with each other, right? So as a part of those fighting process, one will try to kill another, definitely. So it is also happening in our society as well. We are not really killing each other, but we are definitely trying to improve ourselves better so that we could actually give a more impact to the job market these days. So in bacteria also, this resistance phenomena emerge in this fashion. Like one bacteria secretes some metabolites which might not be so detrimental for them, on the other hand, which might be detrimental for the near vicinity cells, so that it could actually kill the surrounding cells and it could start proliferating its own progeny in a very
Mm.
rapid fashion. So now, to answer your question, antibiotics could be anything. It could be, per se, for example, it could be a metabolite product, it could be some extract from a bacteria, it could be a toxin also for a bacteria, but it's not
Yeah,
really other like toxin, not for itself, but for the others. So I wouldn't dare to say it has to be a glycoprotein, it has to be a protein, it has to be a carbohydrate. It could be almost anything. So the one that has an effect to the other cells, not its own cell, it's called basically antibiotic. So it's a
I see.
product from the bacteria.
It's
That's
just a metabolite,
exactly, it's just a metabolite. So you
and
could
it
just
could
put it
be
as a
anything.
it's
Yeah.
a bacterial metabolite,
Okay,
secondary
okay,
metabolite
okay,
to be
okay. So
precise.
now I understood what an antibiotic is, and
Yes.
now what I feel is, as you very correctly mentioned, and in a very simple way, that
Hmm.
if you want to grow, if a bacteria wants to grow, it will develop some toxin or
Hmm.
maybe venom or whatever metabolite it will secrete and then
Hmm.
uh, kill the progeny around, which is a different progeny.
Exactly,
Toxin.
penicillin. Yeah, yeah, yeah. I totally. It's like secreted by a fungus and it kills bacteria. Of course, it's like a
Yeah,
intercommunity fight, but
yeah.
sometimes it's also like now if like okay, fine. I'm not really going into that details. It's like a type six secretion system. Also, it fights with one
Uh-huh,
bacteria and another. So it's not an antibiotic, by the way. Okay, maybe we can discuss this later if you are interested.
uh-huh, uh-huh.
Yep.
Yeah. And my second part of the question is that what can we do to halt this mechanism in bacteria, especially that is you said that a bacteria develops a defense mechanism which is
Mm-hmm.
called as AMR right now.
Mm-hmm. Mm-hmm.
So then you talked about majorly two to three these defense mechanisms. So are there any prevailing processes which
Mm-hmm.
halt these, or how can we inhibit this defense mechanism? In the bacteria.
Of course. You the first thing you can do is you don't really use antibiotics. You don't overuse it, because if you are using antibiotics knowingly or unknowingly, you are selecting for the resistant variants. We are the big believer of Darwinism here at the institute, and we also think antibiotic resistant bacteria are already present in the environment. And if we are giving antibiotics on a very rapid fashion, we are selecting for those variants, those has a resistant cassette. And I will actually demonstrate you in my next presentation, the moment, just the moment. But now what we could do, we have to act smarter than a bacteria. So how we could do that? We have to really think of how the way they are developing resistance mechanisms. And if we could actually block those mechanisms, it doesn't necessarily have to be an antibiotic. So we should definitely go for something called as more sustainable approach. You can go for phage therapy. You can go for like microbiome modulators. You can create some of the products that in turn, disrupts the signaling system between two bacteria. For example, there is a process called as codon sensing. It's a mechanism by which one bacteria communicates with other. And we know now there are some compounds, whether when it comes to gram-negative bacteria, it's called acyl homoserine lactone, and when it comes to gram-positive bacteria, it's like a peptide. So if you know the structure of peptide, and if you could design something which actually go and damage your peptide, or just go and mask your signaling molecules, so that you are breaking the chain, you are breaking the connection between two bacterial cells. So the number one thing, you use less antibiotics. For example, the patient who had gone through a surgery, and they will die if you don't give antibiotics, because in an operation they will have to cut the, like they will have to make some incision, and they will take out your stuff. And in a hospital, it is quite obvious that you will come across a lot of nosocomial pathogens, and they have a freaking amount of resistant genes in it. And then at that time, you are also immunocompromised. Then in that case, antibiotics are necessary. But for example, we started out today's conversation with common cold. Even if you today go to a doctor, I'm not really complaining about them, because they also do not have any options. Because if you are going to a doctor, and if you're saying, I am sick, give me something, and if your doctor is telling, I have nothing to give you, the patient will not simply visit them. So it also has to be very much multi-stakeholder approach. They should also understand. They should Education is also important. You need to educate people, like when you should take antibiotic and when you should not touch even antibiotic. And sometimes, like my mom used to say one thing, it's like a bacha goli. It's like you give something in order to convince them. It's like a placebo effect. You give a sugar candy and you say this is a drug, and if you somehow think psychologically, okay, this is a drug, so if I'm having it, I'll be cured. So that will also sometimes work. So you definitely do not know how it works, but yeah, this is also some of the placebo effects that we could also see. So the one option is you use less. The another option is you design something non-antibiotics, so you could also block the virulence factors. The third option could be you somehow modulate its genome or engineer its genome such a way, maybe you could have them passage for a several generation, so that a pathogenic bacteria may turn out to be a common cell. For
Mhm.
example, if a pathogenic bacteria is turned out to be a common cell, in that case there won't be any harm. Because they might also be an opportunist, they might also resides in your system, and whenever you are immunocompromised, they might start kicking out the infections again. But it's highly impossible, I would say, that if they once become common cell, they will again go for an evolutionary transition and go for a pathogenic phase. It will never happen once they are common cell, unless or until you are immunocompromised. they may not
Mhm,
really trigger
mhm.
those phenomena again.
I see.
These are the three
So,
approaches.
so
Yeah.
I think that this questions Dhara's first question, that could we give antibiotic after the halting mechanism? That first you can halt the mechanism of bacteria and then treat it with antibiotic.
I
So
think that that would be a way more smarter than anything else one could actually even think of.
yeah,
The first,
yeah.
and we experimentally demonstrated at the Schulenburg lab, like sometimes you could actually disrupt the virulence factor first,
Uh-huh.
because you are actually targeting, like basically now I am studying this particular direction. Like my whole research revolves around how an antibiotic resistant bacteria could equally be virulent or avirulent. Because one thing also we still do not understand, whether having an antibiotic resistant genes makes you more virulent or not, because these are two different phenotypes. One is antibiotic resistance, rather antibiotic resistance doesn't really confer to be a virulent. Even if you are antibiotic resistant, you don't really have to be virulent, because already you are so smart that no one can kill you, because you know that I have all the resistant genes. So why should you compromise your host? For example, COVID pandemic, when it started, as you could see that it was so fatal and it created a massive havoc around. It killed a lot of people. But nowadays in Germany, I don't know about you, Tharia, and your place like Freiburg, but in Kiel, we already got some reports from the COVID recently, and they are just giving like a symptoms like a flu. So now, what an evolutionary biologist would say that now COVID is also trying to assimilate inside us, and maybe it may not really so long that it will also become a resident member of in our body. So that is how evolution works.
Mhm,
So,
mhm.
yep.
So I would say bacteria is evolving faster as compared to our brains.
Ah, yeah,
Even
I can
yeah,
understand. Yep.
bacteria evolve faster, even more than our thoughts run. So even
Definitely.
so
No,
more than our
our
thoughts, I
thought
would
runs more
say.
faster. It's like
Yeah.
Mumbai to Manhattan 1 second. So yeah, but of course, I would say any other organism, because the generation time is, you could think of, it's like 20 to 40 minutes.
40 minutes. Yeah.
For example, if Gregor Johann Mendel has started working with bacteria, he could have discovered more, because he decided to work with a pea plant that really made him to wait for 1 year to get a new identical genotypic traits. On the top of that, that guy actually didn't know about genes. He just
Yeah.
determined them as a factor. He
Factors.
didn't even know. Maybe if he could have done all this facility, what we have these days, like NGS and all, he could have given a tons of things. I feel it feels very pity, because we are also in an era we have almost every sort of facilities, but still I think we are not really doing good enough for our
Yeah.
research, because we
Correct.
have NGS and all. But those days, you could think of the area, and also like the audience, I would say, like, well, like they had nothing. Even the
And still they did
microscope,
much better.
they still did much better. The country next to our country is France. Louis Pasteur, he started discovering a lot of
Yeah.
pathogens, man. And now
Yeah.
it is no one discovered. We struggled a lot for a virus, COVID
Yeah.
pandemic.
Yeah.
Even these days, we still don't know the cure.
Yeah.
There are some maladies like cancer, AIDS. Still, we don't know how to cure them. So
Correct.
sometimes it feels like very guilty to say, but it is the thing. Like we are not really so smart. We really need to
Yeah.
work hard. We really need a lot of research to do it.
Yeah.
So, yeah.
So what I personally think is that, see, there are certain species that are already present on this Earth planet,
Mm-hmm.
and if you try to kill them by
Mm-hmm.
certain antibiotics or any
Mm-hmm.
drugs, then it's definite that, as you said, no one wants to be killed or no one wants to die.
Mm-hmm.
So they will generate certain mechanisms to overcome that. So I personally feel that maybe
Mm-hmm.
they will be evolving even faster in few, I would say, few more years ahead, and maybe the life cycle would also change for bacteria, because right now we have so many antibiotics and so many drugs available, which also focuses on the cell cycle arrest and things like
Hmm.
this. So
Hmm.
I personally think that it could be, it's not under our notice right now, but it could be possible that the bacteria's dividing rate would increase, or the time generation time would shorten down,
Hmm.
maybe for 2 minutes, but like that 2 minutes is also a huge, if you
Exactly. Because
if
you
you
never
compare a
know,
E.
in that
coli
two
class.
minutes, they might secrete some virulence factor which might be lethal to yourself. So
Yeah,
if you could
exactly.
shorten it up, that
Yeah,
will
yeah.
definitely help you.
Correct.
That's right.
So I think this answers the Dhara's second question that
Oh,
she
really?
asked,
Wow.
that like you are talking about mechanisms of resistance. So
Hmm.
bacteria can develop other mechanisms of resistance after destroying the first one.
Hmm.
Is it possible like that? Yes, of course, I would
Yes, of
say.
course. Yep.
Uh, or bacteria has more than one
That
resistance
is also
species.
true. Sometimes
Yes.
bacteria has a lot of mechanism to work
Yeah.
on because it never knows what kind of drug is coming. So they are
Yeah.
all ready with every sort of weapons.
Yeah.
And this is the most fascinating thing about bacteria, Dhara. It's like they have a fascinating mechanism called horizontal gene transfer. So if I have
Wow.
something, so I can definitely give it to you
Maybe you can
immediately.
now, since you have just touched this topic of horizontal gene transfer, maybe you can describe it in simple words, that what is horizontal, because I studied it long back in my bachelor's.
Oh God.
So maybe we can
I shouldn't
refresh
take
it.
any new terminologies here. Ah, great. So the first thing is like, uh, there are two transfer mechanism. Like the DNA is getting passed on to the next generation. It's like a vertical transfer that we have like replication and all those like very trivial process, which is also necessary for a cell to carry on. On the other hand, what we have in case of bacteria and some of the fungi is like they try to share the component via a process called horizontal gene transfer. So it has basically three proven mechanisms. There are also some other hypotheses. I wouldn't say that they are proven mechanism, but the three are the most proven mechanism. One is the transformation, where a bacterial cell will actually give a direct DNA molecule to its nearby cell. But there also has to be some condition, like the nearby cell has to be competent one to receive that. Like if I'm giving something to you, you should have that capacity to receive. So if I'm giving some knowledge to some people, I should know their background first. So it is also important, or else नहीं तो भेजा के ऊपर से जाएगा. So it's like this is the first concept called transformation. The another part is it's a very independent one. It's called the transduction, where a particular segment of your DNA are getting transferred to a next bacteria via phage virus. So we call it bacteria phage. It's kind of a virus. Don't confuse here because bacteria phage is not a bacteria, it's a virus, and they are killing bacteria. So what they are doing basically, they're infecting a bacteria, and they are also very specific to a particular strain. So they would try to reassemble their product the moment they attack the bacteria. So sometimes what happens, they try to get the wrong piece of DNA inside the cell. As a result, the moment they have this wrong piece, they would just go and try to sit next to the other bacteria. And the moment it infects, it knowingly or unknowingly, they are transferring a piece of DNA to its nearby cell. The third mechanism, which is more controlled, I would say, and bacteria also knows how to deal with that. It's called conjugation, where this plasmid, there's back in bacteria, there's something called as plasmid. It's a kind of an extra chromosome on DNA. So mostly what we could see these days, like all of the resistant elements are present in plasmid. So for example, if I know that there are some enemies, and those enemies are nothing but antibiotics, and if I want to make my neighbors aware that you should get yourself protected from those enemies, I will share my plasmid with you via conjugation. It's like a tube, two bacterial cell, they are trying to form a tube in between, and as a result, one plasmid going to percolate from one tube, from one bacterial cell to another through that tube, and that gives other one the plasmid. So it's like one has something special. After a few moment, the other one, maybe 1 to 2 hours, the other one also has the same. So it's just copy paste mechanism.
Yeah, I mean, I would say this is something that we had during our bachelor's, and I
Yes,
also did
definitely.
transformation. Everyone does transformation,
Yeah.
transcription,
Hmm.
conjugation in their labs as well.
Hmm.
So I think it was a very brief mechanism to it, right?
Just to also clarify, the microvesicles is also a mechanism, but it's not
Uh-huh.
well established. Microvesicles is not.
Oh,
It's also a mechanism, but it's not well established, and it hasn't
mhm.
really been worked out. So the scientific
But
community doesn't really agree on it. But yeah,
mhm,
this could also be a, like where you throw DNA and the other cells are capturing. It's like a
uh-huh.
more signaling. It's like a Wi-Fi,
Yeah,
something.
yeah,
You have a transmitter and you're
yeah.
transmitting something, and nearby cells are actually trying to gain out of it. So yeah,
Yeah,
but
yeah,
this is not really proven.
vesicular, yeah, 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 yeah. I would say vesicular trafficking, or I don't
Ah,
know
got it.
how it is called in bacteria,
Hmm.
because I am not a microbiologist. I'm a molecular biologist
Yeah.
and a cell biologist. So I deal
So
with
I think it's different
the
from
cells. I
your
deal
classical
with
study.
mice. I deal with human cells.
Mhm.
So this is a very uh, well known and well established mechanism in the mammalian cells, that they do
Mhm.
really communicate with each other through the vesicles. But I'm also not sure about bacteria, because I've never heard of vesicular trafficking between bacteria. And
No,
it would be really
yeah,
interesting if someone wants to do PhD in this topic.
why
And
not?
yeah,
Mm,
because it's upcoming. Okay,
because yeah.
so
Mhm.
we have a question which is a bit immunological. Maybe I could
Oh
have
gosh,
joined for the conversation in this.
gosh.
Interferons are released by bacteria to alert nearby community
Mhm.
when attacked by virus.
Mhm.
Similarly, are there any such mechanism after antibiotic treatment? Like, so he's trying to ask. Thanks, Gaurav. This is a very good question. So he's trying to ask that if you treat a bacteria with certain
Mhm.
antibiotic,
Mhm.
is there any mechanism by which bacteria alerts the other bacteria in surrounding? Because this happens in the cells, especially in the mammalian cells. I don't. First of all, I would like to ask you one
Mhm.
question,
Sure.
Surajit. Does bacteria releases interferons? Do bacteria release interferon? I'm really not sure. Sorry.
I'm also not sure, but obviously they communicate with each
They
other in
communicate,
forms
yeah,
of,
but
yeah,
I don't
in
know
forms of those
interferons to
acyl
be like,
homoserine lactones.
yeah,
So that was
interferon.
my first point that I wanted to say is that, but that's a
Wait.
different
I'm
interpretation.
not done. I'm not done. So,
Oh, sure.
so Gaurav wants to ask is, so first question was, do bacteria secrete interferons? I don't know. And the thing is that he wants to ask is that are there any mechanisms by which bacteria alerts the other community members that, hey, there is something which will kill us, so start developing antibiotic resistance now. So
Yeah,
is there any mechanism?
I just did a Google search honestly because as I'm a PhD student, it's not really possible for me to
Yeah,
know. But I don't think they secrete any interferons, and also
yeah,
I don't think it's a mechanism that is also necessary for them because they already have some alternatives. So why they would go and mimic an eukaryotic cell that is much more
yeah.
complex, right? Because they are very simple and they're happy with that, and they are way more smarter than us. So why they just would like to complicate their life? So they won't do that. So now coming to your question, they have some memory. Honestly, it actually sounds very like sometimes it's very vague because how a bacterial cell might can also have a memory because it has no neurons, it has no connection, it has
Uh-huh.
no signaling procedures which actually makes them to okay, the antibiotic that I got an exposure last year was carboxycillin, so I'm now I'm now somehow protected for that because sort of I have a resistant cassette in it. So but they do have, but we are not really sure how are they getting stored. Even a few days back I had a discussion with some of my friends, and we thought epigenetics could also definitely play a role here because if you are not really storing the memory in a form of DNA, but bacteria has no other choice. It is so poor, it has only DNA, RNA, and all the stuff. It has no histone modification parts and where it could actually go and wrap around like in eukaryotic cells. It has no histone H2, H2A2B, all those domains. So the only kind of methylation changes it could make in its DNA content, and that might sometimes help them. For example, you have a resistant protein. You know this protein is necessary for you to give you protection against any beta lactam antibiotics. And if you have made a methylation profile, and if you made your active site, for example, your active site structure was something like a moon shape, and you made a methylation here, as a result, it's now modified. So you could actually target a more specific antibiotic. So that makes you more fit into the particular surroundings. So now to answer your question, it definitely they sense something in terms of their previous exposure. As a result, they try to manipulate their genome, but they do it whenever it is absolutely necessary for them. Because what we also observe, all this resistance, the moment they acquire this resistant genes, they really need to pay a lot, because if the antibiotics are missing from the environment, they are growing very slower than, because they have already modified them into their genome. But if you, for example, have modified something to your genome, and if you really do not know why did you modify, because now the reason you modified it is not there present, so there is no function of it. So then those kind of compromisations are also happening in bacterial cell. As a result, now people are also switching in this direction of epigenetics. But as an evolutionary biologist, we are somehow try to focus on genomic functions, because
Yeah,
we are believer
yeah,
of Darwinism and genetics,
yeah.
classical methods. So
But I think your answer was quite good. But I think it doesn't answer this question about how do bacteria communicate with each other
okay,
when
sure,
there is an
sure.
antibiotic
I'm sorry.
attack.
I'm sorry. Yeah, in that case, I would say codon sensing is the method. You
What is
alert your
quantum sensing? I, I,
okay.
I
Sure,
know a bit about it, but I
sure.
think audience might be very
I think I
nice.
just gave a quite glimpse of it before. It is like a signaling molecule, or it is sorry, it is a basically it is a signaling system.
Hmm.
By processing it or by doing it, one bacteria alerts the new bacteria, or it actually informs the other bacteria there is something, very dangerous coming. So we must protect ourselves. Let us form a biofilm. So you know about biofilm. So the biofilm is something, it's a mesh-like structure, and it allows them to get a protection against any infections. Now there is also a controversy. People say sometimes, and now it is somewhat clear, like in a biofilm, there won't be any single bacteria inside it. So it's like a mesh structure. You form a very rough, rigid matrix, so your antibiotics may not really penetrate throughout it. So if you could make that, so that is also happening as a function of quorum sensing. So the quorum sensing, as a very simple term, I would say, I came to know from someone that there is a potential danger. So I will just call my friend, and then my friend will call some another friend. They will call some, and then we will try to build up a team, just to in order to, like, in order to not get affected and for that particular stuff. So we would do that. So this is called a quorum sensing mechanism, and it is also very, 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 very important for both gram positive and gram negative organisms. So yeah, to answer your question, I think they would try to alert others by using this mechanism of quorum sensing.
Mm-hmm, mm-hmm.
Yep.
Okay, I
Mhm.
see. I see. So, yeah, Dara says that bacteria are like AI adapting their destroyers' information by evolution for saving their lives.
Bacteria are like artificial intelligence, AI adapting. Okay, yeah.
I
Then this,
think
what is AI adapting?
artificial intelligence, I guess.
Okay, AI adapt
AI,
their destroyers information. Destroyers. Yeah, they could actually destroy the information quite fast. That I would say. For example, you give an antibiotic,
yeah.
they are really outsmarting it in a
So,
fraction of like one or
yeah.
two days. Yeah.
So
We
I want
we
to
do that.
ask you another question
Sure.
on this, that since you were touching the methylation pattern
Oh
and
God.
things like this, No, I will ask you
Yeah,
about
sure.
evolutionary per se.
Hmm.
So what I want to ask is
Hmm.
that don't they forget this memory, especially now? Consider that there is a beta lactame, and the bacteria has a strategy to produce the beta lactamase, which is an
Hmm.
enzyme which cleaves this antibiotic.
Exactly.
So, so now considering this example, now if we remove the pressure, we stop using this antibiotics, right?
Hmm.
So will bacteria go back to their initial uh, genome? See, because logically speaking, it's a tiny organism,
Hmm.
and it will also try to eliminate the junk, because the continuously we are developing new antibiotics, and of course there are gene manipulations happening through various techniques in
Hmm.
bacteria that they are
Hmm.
adapting, so the genome is continuously increasing, you
Hmm.
know so what do you say that if we stop using antibiotic, will they again like just give away this extra DNA, which is now not needed to them because we are not using any antibiotic? So do they forget? Do they forget? Do they have a,
Yeah,
I
any
mean,
mechanism for that?
forgetting memory? Yeah.
Exactly. What nowadays there is a speculation is like bacteria try to modify their genome whenever it's truly necessary for them. If they feels like you attacked some of the very essential component, for example, you have given an antibiotic which targets replication, for example, that's a life saving, that's a process which is really necessary for them. So in that case, they would not really think much about it, they will immediately start mutating because they need protection against it, because they really don't want you to modify or they don't really want to die. As a result, if you are targeting some of the essential life components of a bacterial cell, they would modify it immediately. So those kind of mutations, I would say, would be more genetically assimilated. But the moment it comes, like you are trying to destroy the outer permeable membrane, for say, in that case, they might try to make it more bulging in or bulging out by doing some epigenetic modification. We never know that, but how they do. Uh, of course, now to answer your question, whenever it comes to like they already modified something, how much time did they take to start losing it up? I think the way they develop resistance, it also plays a role here. If they start developing a resistance way faster than anything else, then they would also start losing it way faster than anything else, because it's like a balance between these two, right? So it also depends on a population size. If because in a bacteria, you can't really consider as a single cell. We call it as a colony forming unit, but in a single colony, there are a lot of members in it. For example, you do not know how many millions of bacterium has you got over there. So, for example, some of the particular bacterium, I'm telling it as a singular entity, bacterium, because some of them are hyper resistant and some of them are not really resistant. But that makes them a colony forming unit. And if they feel like that is sufficient enough to give themselves protected against the surrounding, they might not really change themselves a lot there. So then they would try to transiently modify themselves by doing some epigenetic changes. So I would still say epigenetic changes are there. We try to believe that, but if it is still there, it may not really outcompete genetic functions until or unless it's really necessary for them, they will go for genetic modification because they have a short lifespan and they try to modify quite faster than any other organism. Yeah.
I see. I
Hmm.
see. So we have questions. We would be taking them at the relevant time. Please
Sure.
keep posting your questions in the chat box. I will be taking it up on a relevant times. Okay.
Sure.
So, Surajit, now talking about evolution,
Hmm.
right? So the most exciting mechanism of that bacteria developed, uh, already had or developed. I don't know. You should answer this question. But we learned from bacteria is CRISPR-Cas9,
Hmm.
right? It was already there in bacteria, and we learned that okay, we can do gene manipulation through
Hmm.
CRISPR-Cas9, and bacteria could just do it very easily.
Hmm.
Can you say that how bacteria evolve through Casper-Cas9, and how do they generate antibiotic resistance, especially through CRISPR-Cas9? Is it they are related or something?
Oh, I think we need to build this into the context first. Like, it is something bacteria has generated in order to get themselves protected from a bacteriophage infections, right? So if you are giving antibiotics to them, what we speculate these days, you could try to identify those particular protein or the gene segments, and you try to make it out. You try to overexpress to other system, and you could actually use them to give as an alternative. But the problem there, those are DNA elements. And as I told you, the DNA are very sensitive and they could modify, they could do mutation at any point. So even though CRISPR-Cas9 is a very smart way to give protection against any particular antibiotics, but I don't think it's more reliable, and also it's like the genetic function of it also gets quite more hyper mutatable. So it's not a good way out of using as an alternative way out, I would say personally. But I'm also not really expert in that, to be honest. But yeah, I think apart from CRISPR-Cas system, I think the phage therapy has more promising results because it's somewhat strain specific. Even though there are some claims that bacteria are sometimes phage resistant, so they are not really dying for that infection. So now the fundamental is whatever you could give to bacteria, they will try to develop some mechanism to it. It is like a kind of Newton's third law. Every action has opposite and equal reaction. So if there is an effect, you will expect a side effect. So this is the fundamental of evolutionary stuff. Like, you can't say anything about, like, for example, I'm not against any Ayurveda or any product, but I don't believe that as a alternative medicine because they don't know how their drug works. But on the other hand, if you could think of our rational drug discovery approach, we know this is the medicine and this is the way it works. And also we know these are the side effects. And as a result, we are giving you the other drugs to treat those side effects. But in case of other alternative medicines, I'm not really sure
Yeah,
how are they working. So in that case, how would I
yeah.
predict their side effects?
I see. So I would like to take this chance and tell something that what usually we do. So because I was working for some while in a lab in Germany on this phytochemicals
Hmm.
and immunity. So what
Okay.
we see is this phytochemicals, that is how the Ayurveda or homeopathic or these medicines, I would say, it work usually is through innate immune system. So Kruti also asked this question. So Kruti also asked this question that you said that we can try to weaken the bacteria, right?
Hmm.
But somehow is it possible to strengthen our immunity to deal with it? So somewhere I would say the Ayurveda and homeopathic and these medicines, they usually target our innate immune system. So
That's
it
good then.
tries to, yeah, it tries to activate your TLR. I mean, it tries to activate your interferon or TNF alpha or some non-specific immune, I would say, immune regulations, uh, which also signals to a more
Hmm.
specific kind of T cell and B cell response. So somewhere, yes, they do really help, and developing, like strengthening the immunity is also important somehow. But it is something really, I would say, innate immune response and not a targeted
Okay.
therapy towards bacteria, I would say.
Then I
So
think it's a good one because no, my understanding with
yeah,
Ayurveda was on the other context. It's like
I
the
understood.
mechanism. The mechanism I don't still know what how it's
Yeah.
working. So if I
No,
don't
no,
have
no.
any,
It's it's clearly understood. It's not known because it's a dynamic immune system.
Okay.
And
Yeah.
if you try to give some drug, or which is a very non-specific drug, and
Yes.
you don't know, you have to just shoot somewhere that where does this target, it's a lot of immune response that goes on. Like the
Exactly.
TNF is activated, interferon is activated, IL-17, IL-2, IL-1, everything is elevated. You
If
never
you
know
could, if
what
you
goes
could
on.
boost up your immune system, you don't need any
Yeah.
antibiotics or any drug.
Yes.
That's for sure.
Yeah, exactly. So it could be done by simple exercises as
Exactly.
well.
Very safe.
And recently, I would like to I would like to tell this, since we have
Mm-hmm.
now touched a bit of
Definitely.
immunology. Recently, we had a journal club in my lab,
Mm-hmm.
and so we were basically discussing about cell death. Okay,
Mm-hmm.
that how cell dies, and then there was an immune perspective that nowadays there are endogenous retroviruses which
Mm-hmm.
are also involved in giving you resistance towards bacteria. Especially, there are certain antimicrobial peptides that are on your skin. I'll give a very classic example, Surajit, and I would
Mm-hmm.
like to ask you a question
Sure.
based upon that. We have a lot of antimicrobial peptides on our hands, on
Mm-hmm.
on our in our mouth, or
Mm-hmm.
our epithelial linings. You know,
Mm-hmm.
why bacteria which are already present, like S. aureus, is already present on our skin.
Mm-hmm.
And we have antibiotic peptides for this, right?
Mm-hmm.
So why does this bacteria do not develop AMR? That the antibiotic peptides that we are producing naturally from our body.
I think here we need to distinguish a little bit about what are our resident microflora and what's actually the pathogenic one. Like,
So they works on the symbiotic relationship
exactly,
between together,
exactly.
and that's what I wanted to point out, that it's a symbiotic relationship, and there are certain endogenous retroviruses which
Mm-hmm.
are actually helping this to develop a symbiotic relationship between gut microbiome and humans, as well as the skin microflora and the humans. So I'm not very sure, but it
Mm-hmm.
was a really good paper.
Oh, that's
Maybe,
great.
yeah,
Maybe
yeah,
you could,
maybe you
yeah,
can.
you could just
can
give me the link or something.
I will definitely give you the ring. So there, but what I was surprised is they didn't anywhere talk about evolution of bacteria.
I
And
think
I was like shocked that the main factor that plays role in antibiotic resistance is evolution
yes.
of bacteria. But in whole paper there was no, and it was a paper of cell.
Oh gosh, it's somewhat understudied, but luckily it's been now rediscovered after this COVID pandemic, and now people are
Yeah,
also focusing. And to be honest, at the place where I belong, it also has a strong support for evolutionary research because of an institute like Max Planck Institute of for Evolution, because they are funded by federal ministry directly. And
yeah, yeah,
so, but when it comes to India, I would still say it's not it's kind of a dogma these days. We are not really focusing on fundamental research. Even I
yeah,
also have an elder brother. He's also working at IIT Delhi as a postdoc, and he I had a discussion with him a few days back, and he said, you know what? These days, our research is so much getting commercialized. If I'm not going to give you a product at the end, I'm not a good researcher. So this is not the case. I shouldn't say if that is the scenario, I will never return to India because I'm researching on some fundamental question and I do not have any idea how to marketize that. Maybe like friends of mine, like as I already told Ankit, he's working at IIT Bombay. He might know better than me how to structure it in a more commercial way
commercialize.
because I don't know how to sell my research. Honestly,
Yeah,
I know how to sell my research to get a postdoc somewhere, but I don't know how to sell my research in the market. So that is a
yeah.
yeah.
I understood. I completely understand this. Even same is the same issue is with many of the people who are working with gut microbiome and stuff like this as well. It's quite complicated right now. So Gaurav is asking that
Mm-hmm.
ideally, I think you could be correct, and you could interpret it in a correct way because the terminologies are I
Okay.
would say
Okay.
microbiologist would
Gaurav,
understand.
Somalia.
So ideally, AST must
Okay.
be
Yep. Mhm.
performed to treat infections, but
Mhm.
in reality, these tests are not performed during treatment of patients.
That's a good point. Exactly. Yeah.
Also,
Mhm.
CLSI standards are not followed to prescribe the antibiotics, and higher order antibiotics are prescribed, which are not necessary for that individual. Okay. First, let me understand this question. What is AST?
It's an antimicrobial susceptibility testing. We say it,
Okay.
we call it antibiogram in short. You have got an infection with a bug called Pseudomonas epidemic. So you got an
Hmm.
infection with a bug called Acinetobacter baumannii, and you want to
Hmm.
know what are the resistant cases present in it. So what you would do is like you for your infection is in the nose, you would get them, and you just immediately you go for an antibiogram profile. You have them plated on an agar plate, and you just put an MIC strip or an E-test strip. So the next morning you get a results on resistant genes. Like these are the resistant cancers that might be present. Now the CLSI, it's like it's like it's an institute which says like which is the abbreviation of it would be Clinical Laboratory Standard Institute. So it sets the guidelines for like this could be the MIC values, like the minimum inhibitory concentration. So up to this concentration, bacteria can actually withstand the antibiotic pressure. So after that, like there won't be any like lethal effect. So like the minimum inhibitory concentration is something called as MIC. I'm sorry, but I just abbreviated the term. I could have expanded it before. So it's like what is the minimum amount of antibiotics that one need in order to like get themselves to be like killed by that antibiotic. So yeah, this is the
Hmm.
MIC. Yep.
Hmm.
So
I
now
see.
to answer your question, ideally AST must be performed to treat infection. Yeah, that is a mistake that we often do. Even that is
Hmm.
the reason why we still do not know if an infection is growing inside a patient. For example, there also those bacteria, those small superbugs, meet some other resident microbiota, and they could actually make them virulent.
Yes.
Gaurav. Like the problem is if you have a superbug and you got a superbug from superbug means I'm sorry again I'm using a terminology, it's like a bacteria when it is multi-drug resistant we call it superbug because it's like super strong. It's a bug and it's like super, and it has almost every resistant cassette in it. Like cassette means gene, right? So in that sense I would say there is a potential damage what we do in a patient treatment. We definitely do not care about those MIC on an interval basis, but it would have been really nice to see. But how many times you would ask your patient, give a sample and I would do an MIC and then I will treat antibiotics. As I told you very first, if your doctor, I'm going to ask you a question to you, Gaurav. If you are going to a doctor with a symptom and if you say, doctor, I have this, what would you reckon? Your doctor will prescribe you antibiotic, or if your doctor will prescribe you something, or instead of that, they will say, I can only give you something if you are coming with this report. Then you will say you have no knowledge, you don't know how to treat your patient. Then you will only say your doctor, you don't know how to treat a patient. So instead of that, what they are doing, they are blindly guessing something based on their understanding and experiment. Okay, this is a flu season, probably. Okay, this is a, and as I already said, some of them are also mediated by virus. So doctors are also prescribing because they don't have any other options to treat with. There was a saying from some of my school teacher when we were doing
Yes.
like
Yes.
our schooling. He said, uh, if you take antibiotics, a viral infection takes 1 week to get cured. But if you do not take, it just takes 7 days. So that means it doesn't really make sense. 1 week and 7 days both are similar, right? So,
Yeah. I
but I
see.
still think there needs to a lot of things that we need to do in rapid testing. Like
Hmm.
you give a sample, immediately you get a report, and then your doctor knows, okay, so these are the resistant genes that
Maybe CRP test,
CRP test is something immunogenic test that you could,
immunogenic test,
yeah,
not for
know about
bacteria.
the status of the patient. You may not really know about your bacteria, but you know,
Yeah, exactly.
yeah, so the bacteria is Somalia. Yeah, so sorry. So yeah, you need to know about your bacteria first. So in order to do that, people are these days what they are doing, they are using Raman spectroscopy. It is also a fascinating thing.
Oh.
Raman spectroscopy, what it offers, it gives a distinct spectra for a single sort of antibiotic resistant genes. If you have, for
Mhm.
example, antibiotic resistant genes for beta lactam, so if you have certain beta lactams, for example, there is a variant called SHV1, there is a variant called CTXM15. So if you have a CTXM15 cassette in it, the spectra look like this. So that is very simple. If a doctor has a this machine like Raman spectrophotometer, like it's a basic spectrophotometer, but the fundamentals are different. So only if he could, or he or she could take your sample, run through the spectrophotometer, and he will get the data. And then he could say, okay, so you are already resistant to this drug. So I have only these two options to treat with, but I will not go for these two options because this is something I will keep for the last minute. Instead of that, I will say, you have this microbiota. These are the probiotics. You have it. You do your other stuff. Don't rely on antibiotics. You will be all fine. So if he
I
could,
see.
do
I
that, I
see.
think it's more better.
So Somila is asking that, and even Dhara has similar kind of question. Please, maybe we can keep this short. That can we somehow target the communication between two bacteria? Then could we solve this AMR problem? Like if a bacteria do not communicate with other bacteria, I think it cannot be communicated that okay, I am getting killed by antibiotic. So
Hmm.
what are your takes on this?
I actually still didn't get the question. I'm sorry.
So it's like so when so as we discussed earlier, if a
Hmm.
bacteria comes across any antibiotic, right? So it alerts the community around that hey, I am being attacked by
Hmm.
antibiotic, right? And then it communicates with other bacteria through quorum sensing
Hmm.
mechanisms, other
Hmm.
maybe vesicular mechanisms now
Hmm.
that okay, hey, I'm attacked.
Hmm.
So can we target this
Yeah.
that we stop
Okay.
the
Okay.
communication between bacteria?
Yes, you can definitely do that. There are some studies which has shown like quorum quenchers, it's like it actually targets those auto inducers. That means acyl homoserine lactones, and it could also, for example, you are creating some drugs that
Hmm.
will go and bind to the receptor of your nearby bacteria so
Hmm.
that the quorum sensing molecule will not go and bind there because your receptor has been already occupied. So you
Yeah.
know the structure of your receptor. You
Hmm.
design your drug in such a fashion that
Hmm.
would eventually allow you to
Hmm.
occupy the receptor of your nearby cell.
Hmm.
You are done.
So
Yeah,
so that's how you can also
it
inhibit the
is there.
biofilm
A lot of
formation.
studies are there. Exactly, it is there. And it's a
Hmm. I
fascinating
see.
thing. Yeah.
Okay. Somila, you had some question, so maybe you could quickly ask Surajit.
Hmm.
Yeah, I would like to set some point. You talked about the
Hmm.
a fast detection of bacterial and viral diseases. So I have
Hmm.
come across some means while researching, I come across some techniques which is known as Hudson with CRISPR. So it is used in a non-hospitable technique. Means it what it does is basically it detects the DNA molecules up to femtomolar region, the femtomolar range, and it has a really
Yeah.
high sensitivity. And also, Professor
Just,
Devajyoti
I'm in a meeting.
Chakraborty
I just finished it. I'm so sorry,
from
but
IG. Yeah,
I actually like some of my colleagues just arrived, and I just wanted to say him hello. So I'm so sorry, but I didn't listen. Could you please?
and repeat it again. So there are some fast diagnostics techniques which are developing.
Hmm.
Such one is Hudson-based CRISPR, and which
Hmm.
uses really very small apparatus, just a paper
Okay.
strip and
Okay.
basic principle of chromatography, and it
Wow.
really detects the viral or bacteria, viral DNA up to
Hmm.
femtomolar
Okay.
sensitivity, and
Hmm.
even uh, Professor Devajyoti Chakraborty from CSIR IGIB, he has developed Feluda technique, which is also
Yes.
based on CRISPR as well. So, but
Mm-hmm.
these techniques are basically for viral diseases. And even Professor Amit Singh, also he has got his group has got a patent on such diagnostic techniques using uh, GFP. for viral diagnostics for COVID.
Yeah.
But as of now, what I found that early diagnostics in bacteria can also be developed by using such kind of principle that the our CRISPR DNA, the reporter DNA, gets combined with the bacterial DNA, and it flourishes at it's at really sensitive level. So this kind of research is also going on for bacterial diagnostics.
That is great. Even the moment, actually, I told you before, if we could detect them in a very first stage, we already, the game is on our side. So that is the only thing. The most problem we are doing with the guesswork, because the doctor, they don't know about the bug and they're giving something. So if you know your bug, then only you can design your drug. And then if you make this bug and drug pair, it's fine. So the detection is the key. But unfortunately, we didn't have much good methods to detect all those facets. But I think if we could come up with some more sophisticated techniques, and the one Somalia has explained just now, it would have been way more better. Yeah, I still say, yeah, great. Mm. And in that case, you don't have to be rely on evolution because this technique may not really depend on it might actually develop based on the evolutionary phenomena, but you don't have to think about evolution there. So I think we also have a few questions from Kiran. Like I know Kiranji personally. Actually, I worked with her during my masters, and I'm glad Kiranji joined today. Thanks for joining. So I think she also has a question now. It's a very fascinating topic that you are discussing today. So there are many antibiotics that no more used. Yeah, like penicillins. We also know that plasmid gets lost when not on the selection question exactly. So is it possible that plasmid stability is possible in the absence of? Yes, yeah. Actually, it is. I would definitely ask you to look some of our group. Like it's like one of our collaborator. Her name is Professor PROF Professor Tal Dagan Dagan. She's basically from Israel, but she's living here in Kiel with us, and she is working on this plasmid stability. And she's actually she what she found out. Okay, first let me read the second part, and thus we can get. Yeah, okay. So yeah, you can actually impose a minimum level of selection pressure in order to protect this kind of phenomena. So that might actually help them to get protect themselves from the plasmid curing. So which may in because if once it is lost, it is also really difficult for it to track like where it has gone. On the other side, this plasmid stability is also depends on some other phenomena, like whether it has some essential components or it is only the antibiotic resistant genes on it. Because if it is so essential, bacteria might not really prefer to maintain as a separate entity. Instead of that, they try to internalize that inside their chromosome. So it is also, I think, I would definitely ask you to go through Professor Tull's research. She is extensively working in this domain. So, but I'm not so expert in that. But yeah, this is really happening. This is also possible. Did I answer your question?
So now moving ahead towards the last part, we would love to hear about your presentation. Love to see what you have got today in the presentation. Maybe you can keep it short,
Sure.
and you can quickly summarize on your presentation. So now I would love to see what you present today. What you
But
have
before that,
something
am I
interesting
allowed to
for.
ask if anyone of like I don't know how many of you are from the field of antibiotic resistance? Because if I am going to use some sort of like terminologies which are very much focused in this domain, would that be okay? Like, do you know what are antibiotics? I think I tried to clarify a bit about this, but if you just feel free, okay, if you don't really understand anything about and if you need a further clarification here. So yeah. All right. So I
Maybe
can just
maybe we can ask that who is interested ahead to work towards antibiotic resistance
Okay.
in
Yeah.
future, maybe
Who
for their
really wants to
PhD
work
or
in this
for
domain?
their
That's a
postdoc
pressing
or
issue,
something
and
like
it's
that.
very rewarding if you work with a pressing
Yeah,
issue.
or who already was.
So yeah, who wants to work in this domain?
Yeah,
Please welcome. Please write
you
us.
can raise your hand.
Please write us.
Yeah. Okay, so Surajit, now let's
Yeah.
go
Let's
ahead with
let's
your
start with the presentation.
presentation.
Just a moment. I think I haven't really shared my screen. Now this will be difficult again. Enter screen. Okay. I think I have got it. Share. Just a moment. Can you see something now? Can you see my slides? If any one of you could just say,
We can see. We can see.
Yes, yes,
great.
we can see.
So is it in the presentation mode now?
Yes, yes, yes.
Great. So as I was talking with you guys, so I'm based at Yale University. So we are also funded by societies like TransEvo, which is called as Translational Evolutionary Research. And I love to mention this name before any of my talk because it is only care about translational evolutionary research these days. And we are also funded by Max Planck Institute for Evolutionary Biology in Cologne. So this is the place where I am living at this moment. So coming to my next slide, I would try to bit advertise about this place of northern Germany where we are located at now. So as you could see here, we are actually located at the very further northern part. Maybe I could get a pointer for the sake of, yeah. So we are actually located here. This is the place where I'm living, and this is the Max Planck Institute where it is located at. So sometime, like, for the last 10 to 15 years, due to an increased number of PI doing in this direction of evolutionary biology research, now Kiel has been considered as a hotspot of evolutionary research. We have a strong collaboration with Frankfurt, Leipzig, Jena, and also Berlin to some extent. Some of our other PIs are also working there. So now the second part, what you could see here is like these are the fundamentals that we often think of when it comes to tackling the crisis of antimicrobial resistance. The first one is public awareness, the one that I mentioned you. We don't have proper knowledge. We are not really so much like, we don't have much about it. Like we don't know how to work with an antibiotics and when to take an antibiotics. We often talk about sanitization, hygiene. We talk about rapid diagnosis. Just now we had a discussion. We talk about developing new drug surveillance functions. We talk about funds, mobility, and all. But what we don't care about evolution. Again, I'm highlighting this again and again just to make you understand. Evolution is at the core of impending antibiotic crisis. And if you don't really understand how fast a bacterium evolved, you may not really come up with a therapeutic approach. I will try to convince you by showing this slide, like why do we think like evolutionary concepts can bring about new like direction in research. So what is the problem that we could see these days? A previous lab member, he was a PhD and a postdoc with us. His name is Life Duffers, and he worked with two patient samples and with cystic fibrosis. And if you have an infection, like if it's a basically genetic disorder, but you get a pseudomonas infection when you have a cystic fibrosis. So these two patients, obviously, I didn't disclose their name because of the transparency and because of also the ethical issues. So the patient 2 and 5. So what we could see is the level of resistance development that they were getting. So in the y-axis, you could see the minimum inhibitory concentration are going like from a 0th state to the last stage, like increasing. And then if you could consider about the patient 5, it's also like in a treatment process, the evolution of drug resistance are quite fast. So the one, like I think Gaurav was telling, it is like we try to find it out, like how a bacteria during an infection evolve resistant to antibiotics. So this is also indeed happening in reality. So it's not something that we could only speculate because we have an evidence now. So where could I contribute, or where our team could contribute, is that we definitely did something called as negative hysteresis. Now, what is this negative hysteresis all about? This phenomenon was not really discovered by us. Rather, the idea was discovered by like some of our previous lab mates. His name is Dr. Roderick Rahmat. He also worked with us at the Schulenburg lab. So what he observed, if you give a small exposure to a single antibiotics, you wash your media, you resuspend them in a buffer, you give the another antibiotics. That produces enhanced killing effect. That means you give a short pre-treatment followed by a post-treatment with another antibiotics, and they have to be mechanistically different. The first one you are going to administer has to be different than the other one. So if you could do that, then immediately, for example, you could see pre-treatment with gentamicin, ciprofloxacin, and carbanicillin are marked in red, gray, and black. How it actually, sensitizes the effect of post-treatment. The only exposure we are giving for 15 minutes, like you are suspending yourself for 15 minutes, centrifuge it, wash everything, give it another antibiotics. But the problem is you can't do it with your patient. You can't say, I am giving you some antibiotics, you inhale it, then you throw everything, I'm giving you another antibiotic. But if you could have established it in reality, it would have been really nicer, because this is the way you could actually fool bacteria. Now, now this is something like, why do you believe me? Because if I could show you some figures, I should show you some microscopy images. So see here, this is the inducible negative hysteresis effect. What we could find out is that this is the control treatment that we have taken in time-lapse microscopy at 360 minutes. And on the other hand, we did a pre-treatment with carbanicillin. So the one that has been pre-treated with carbanicillin, it's like they all died, and we did a live staining here with a DAPI. So the other concepts, what we developed is like collateral sensitivity. This is a fundamental of a physics method, to be honest. It's nothing to do with biology. So in physics, what we observe, if a population is resistant to an antibiotic, the moment you switch to another antibiotic, they might be more susceptible to it, because we assume that if your population is gaining resistance against a single antibiotics, the moment you are switching them, if they are extensively resistant on that particular thing, they may not be that much resistant to the other antibiotics. So you are making them full again. The first one, but don't get confused here. Hysteresis is something where we are giving a pre-exposure of very short time. But here I am saying you switch antibiotics in between. So you give first antibiotic and then you give second antibiotics, and that has to be for a longer period. So now let us consider an example here. These are the bacterial population. We are giving yellow antibiotics. As a result, all this sky color, this ocean color, I don't know, it's like bluish to me. So they are also getting died. The moment you are switching them to blue antibiotics, then you are targeting this yellow population as well. Moving to my next slide, as I try to figure out try to show you what exactly are we also working on. We try to generate this database now, so we know what are the drug pairs. Now, in the y-axis, you could say these are the resistant populations. Like, for example, these are the resistant lines that has been evolved in due course of infections. Now, we challenge them with another antibiotic. So what we observed here is like, if you are resistant to ciprofloxacin, if you are challenging them with gentamicin, they are sensitive. So this is how you should look this whole heat map. So this was a work done by some of our previous PhD student, Camilo Barbosa. He's now doing his postdoctoral research. at MIT, and he worked with, of course, my supervisor, and he is my supervisor, Professor Henry Schulenberg. So this panel is really important for us to know the drug pair. Now, this is really unfortunate for us to see. This amount of sensitivity is very less, but the resistance is more. If you are resistant to one antibiotics, you are also resistant to for another antibiotic. For example, ciprofloxacin is also resistant to ceftazidime. Doripenem is also somewhat resistant to like imipenem, but that is also quite obvious because they are same drug class. As I've already told you, if they are two different drugs, it's very easy for them, and it also does make sense, right? And so we could try to do this method in clinical context because cellular hysteresis is something it's very difficult to follow. But we could definitely do it. All right, as I was not really allowed to give a brief presentation, and I just don't want to bore you with a lot of stuff. I'm going to end it here. So this is my lab where I'm working now. Recently had a trip to Portugal, and thanks for your thanks for listening.
Hi, Surajit. It was a really good presentation. I would say very crisp and quite
If you
interesting.
have any specific question, I think Ankit has a
So,
question. Ankit, please speak up.
Yeah. Hi, Surajit.
I
Hi.
Hi.
yes, you
So,
should
yeah,
go
very
ahead.
nice presentation. It was a very insightful one.
Hello.
Uh,
Yes. Thanks. Thanks.
yeah,
Thanks. Thanks for all your words.
yeah. So, I am not much knowledgeable about this field of antimicrobial resistance, although I
Mm-hmm.
started from microbiology, but still
Mm-hmm.
I
Sure.
can see that you have provided a lot of information and would really help all the budding researchers who are going to
Definitely.
follow that
I
path.
would say. Yeah.
So, and of course, happy birthday to you.
Oh, thank you
So,
so much.
yeah,
But no, it's. I didn't want to
yeah, yeah,
disclose
yeah,
anything. So it's fine.
yeah.
Okay.
No
Thank you so
problem.
much. Yeah.
So, I actually I am from immunology background, and
Mm-hmm.
focusing on that thought, revolving around that thought, I have a particular question. So,
Sure.
antibiotics, as you have discussed, that antibiotics have been shown to target various bacterial activities such as nucleic acids and various
Mm-hmm.
protein productions or cell membrane, cell wall, uh, or essential metabolic pathways. So bacteria, on the contrary, they can also develop drug resistance by mutating the bacterial targets that these
Mm-hmm.
antibiotics are aimed
That's
at,
what they're doing.
or
Yeah.
inactivating or
Mm-hmm.
pumping out the drugs, as you said, E plus pumping,
Exactly. That's what
or
they're
even
doing. Yeah.
acquiring resistance genes. Yeah.
Why not?
So
Yes.
actually, several previous reports have been seen, like I've seen, and some papers I've seen some new classes of antimicrobials which are known as
Mm-hmm.
immunoantibiotics. So
Okay.
these immunoantibiotics have been reported to target that MEP pathway, which is responsible for the synthesis of riboflavin in bacteria. So
Okay.
basically, I don't know much about that, but one or two lines I know a basic thing that basically these molecules have been reported to kill the multi-drug resistant microbes, while
Mm-hmm.
also boosting the natural immune response of the host. So I would like to know if you have any views on this, or can this be considered as a potential strategy in the global fight against AMR,
How,
or
how,
where do you
how
see
it
this
works,
coming?
Ankit? Does it target any essential component of bacteria, which is
Yeah.
very
It
necessary?
basically targets the MEP pathway. I think I don't exactly remember the full form. It is something methyl, some erythritol phosphate pathway or something like that.
Okay.
Basically, it is
Okay.
responsible for the riboflavin biosynthesis pathway. So in bacteria.
I think
So
bacteria
what I
suffers
have,
a lot. Yeah, yeah. Bacteria suffers
Yes,
a lot if you target any
yes,
essential
Ankit.
component.
I have also gone through this.
I'm sorry.
yeah.
Yeah. Like, did you say something? Did you say anything that I couldn't follow you?
No, I did not
No,
say
no,
anything. So
I was just supporting Ankit in this that I have also
Okay. Okay. Wow. Then it's good.
I was also
Quite intrigued by the stuff.
I've also gone through the
This paper.
technique.
Okay.
Yes, yes.
Wow. That's great. But
yeah.
yeah, I
So that
think.
is the thing. So yeah. Please. Yes.
Sure. I think if those components are somehow targeting the essential function, which are really vital for a bacterial cell to live in or to thrive in some, then I think there does feel a lot of difficulties there. They do feel a lot of difficulties there, because what we observed like this, whenever we give them fluoroquinolone, so that is something which, you know, which gives a pressure on DNA guidance function. Yeah, so it is also very necessary for them to go keep on with the replication process. They definitely, like, it took a while for them to develop resistance. On the other hand, we gave something called as beta lactam. So it's like, you give beta lactam today, I'm assuring you the next morning you will see the zone of diameter, like, sorry, the zone of diameter is something like, if you have plate your antibiotics, like if you have an antibiotic in your plate and you have already plated your bacteria, then you could actually see some zone of inhibition. Well, that means like your antibiotics are actually killing those bacteria surrounded by. So I think it again it depends on how well are they interacting with bacterial components. If it is very essential, and if it is targeting the pathway the way you explained, which is very vital, then I think it should not be. It should be at least for some years. There should not be any issues. But again, the problem is this is something inevitable. Like, you can't really avoid. Now the company people might actually tell, okay, we developed something, never ever bacteria will be able to develop resistance to it. If they're telling that to you, they are simply shitting, because it is
Yeah,
never
yeah,
possible.
I don't think any company can guarantee that. If they are guaranteeing
No,
that
no
it's
one, no, no
developing,
one will
because
guarantee that.
yeah, yeah, it's
Exactly.
continuously
So
evolving. So
yeah, that is for sure. But it also, what we also found out, like those antimicrobial peptides, it also depends on how big your molecule is. If your molecule is so big, that time also bacteria face a lot of difficulty to tackle with those big molecules. So that also doesn't mean you create some, you just add peptide, maybe like a decapeptide or something that might be a good antimicrobial agent, not like that. But if your molecule size is big, then sometimes it's like stereochemically favorable. We have seen it in a structural biology context. We haven't really evaluated in vitro level, but I would definitely say that we should start using as less as we can, until or unless, for example, as you have been working in the field of cancer biology, like so the patient that has got an operation, like a friend of mine father's, he had a serious heart ailments and he had to gone through a surgery, and after that he actually contracted pseudomonas aeruginosa infection. Like, I'm not saying luckily, but it was luckily for me. Why I'm saying, because I'm also working in this direction. So it was my joy to see how many resistant cassette those bacterium had. So it also depends on like what kind of variants or what kind of superbug are you getting, and also it depends on how many resistant genes it has got. So it also depends a little bit on the context. So yeah.
So,
Yeah.
Surajit,
all
happy
right, yeah,
birthday
I got the
to
point.
you.
Thank you,
Thanks so much.
thank you so much. I didn't want to disclose, Ankit. It's not good that you said, because it's fine anyhow. It's okay. Thanks, thanks.
It's okay. It's nothing to be ashamed of. It's your birthday, man.
Surajit,
There is nothing special about the birthday,
happy
It's
birthday.
thank you, thanks,
Happy birthday, Surajit.
thank you so much.
Yeah.
If I could have told this to
Yeah.
you guys before, you
So,
could have
uh,
never hosted today. Or else, if you could have asked me to do it next week,
yeah.
I'm also very much bottlenecked with my own sort of experiments here at the institute. So I would have never tried to give a presentation. So that is the reason why I didn't say anything. I just thought, okay, bas ho jaye. So then I can do my work. And also, I'm traveling to
How
India.
you enjoyed your birthday with the psycho
Yeah, that is also true. Yeah, thanks, thanks.
and plus YouTube community?
Thanks. Thanks. Thanks for all your wishes. Thanks. I truly appreciate. Yeah. Is there any question that I could actually
Yeah.
help you to clarify
Okay,
this to the
Surajit.
audience? Oh God, I think
I think
all have left.
maybe we can take last question. So by giving the first antibiotic and distract those resistance, so other antibiotic can work properly. Okay. Okay. Let us now come to an end, Surajit. I guess
Don't
it's
call me
quite a
sir, by the
lot
way.
time, 2 hours. I think we can have it again.
Yeah, sure, sure, definitely.
Yeah.
I'm really thankful, guys, that you
So
joined. You
we
made
can
your
have
time
this
for
talk
this.
again. Yeah.
Okay,
So
okay,
what
man.
would be your concluding remarks?
You take
What
antibiotics
would be
whenever
your
it.
concluding
Yeah,
remarks?
yeah. You take antibiotics whenever it's necessary. Actually, in India, it's quite unfortunate that you get antibiotics very easily in the pharmacy. So don't misuse it, because in Germany, you if you go for an antibiotic, you go to any apotheke, like there's a pharmacy in Germany, so they won't give you antibiotic, and they will be very like they will start arguing with you if you say, I don't have any prescription, but I need this medicine. But they are actually giving me, because I also had like this kind of like I got a in sometimes of some infections here in my throat, not in my throat, it's my in my lip. So I just wanted to get a ointment for that, but they were quite flexible with it. They immediately gave me. But when it comes to antibiotics, they are very strong, and they are saying, no, we can't sell it until or unless give us prescription. So I think this also needs to be somewhat followed in India. We need to think of how exactly we could try to manipulate this, because the supply chain can only work if an effort comes at a multi institutional level. Okay, that's it. Thanks for joining.
Okay,
Have a
great,
nice.
Surajit. It was a really great talk, and I think we should have one more live, or maybe one more side chat with you, maybe in upcoming months in future when you have your first publication, maybe, and then we can also discuss about that
Then it may not never
maybe in
happen.
future.
It may not never happen then. It's fine. Okay. Okay. Yeah. That's a good
Yeah,
motivation, by the way.
yeah.
Yeah.
Looking at the mechanisms that the German PI especially follow, it's very
Hmm.
difficult to get a publication, or you get it after 3 years at least.
Yeah. Okay.
So
True.
let's see how it goes.
Okay. Thanks for joining.
Let's see how
See
it
you
goes
around
now.
again.
So
Bye bye.
yes, thank you everyone. So we have a
Okay. 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 Sorry. Sorry. Sorry.
wait.
Sorry.
We have a feedback form for everyone here. You It would be really good if you all can fill it and help us to
Don't call me sir. I'm not
grow
calling
more.
my sir. Like
And
I'm not calling my professor as sir here. So Surajit is fine, or Surajit Bhaiya is also
yeah,
okay.
yeah, yeah. So Surajit Bhaiya.
Bhaiya is also okay. Surajit Sai is
Thanks
the
for
Bengali
the
version. Yeah.
wonderful session today. Yeah, thanks for the wonderful session today.
Yep.
And now we officially sign off
Yep.
for the today's discussion, but we are
Mhm.
not done. We will be upcoming with many more sessions like this. We will be inviting much more people from various fields. So maybe you can write down in the feedback form that you want this topic or that topic, so that we can really
Yeah,
uh,
sure.
get on with the speakers,
Keep in
right? We can
touch.
find the speakers.
Stay
And
with us.
Surajit is also one of our admin member. Yeah.
Mhm.
So
Like the
okay.
SciCom Synapse was an initiative by all of us, but the area started in pandemic when people were
Yeah.
actually facing those kind of difficulties, what to do, where to go, those kind of black boxes. So he started this venture. Now it's
Yeah.
been successfully running for 2 years almost. and now we somehow we tried to change, modify it, make it more attractive. So it was before scientific community, now it's been reintroduced as the psychom syllabus. So we also understand it needs your support, so
And the
please.
name was
And also we need a lot of avid listeners
and
like
the
you.
name was coined by Mr. Surajit.
No, that's like just a random stuff, it's fine, no worries.
And this new name was coined by Mr. Surajit. I
uh,
think
Now it's too much happening. It's like a lot of more birthday gift. Thank you so much for all your gifts. I truly appreciate.
I see. Okay then. Okay
Okay,
then.
see you around.
Chalo.
Bye bye.
Bye bye. Let's
Thank
take a
you,
leave for a
everyone.
short time and we'll be back. Bye bye. Bye everyone. Thanks for joining.